Okay, so this is the pre-lecture for the subspecies level conservation. Sorry for the last minute. We just got didn't have much time between Thursday and now to put this all together. And Jordan and Emily Abernathy, who will be doing the lecture tomorrow in class, were working on their stuff over the weekend. So, and I'm trying to not overlap with them too much. So I'm going to talk a bit about why evolution is important to conservation, the DPS, ESU, and ESA. I'm going to go lightly on that because Jordan's going to talk a bit about it, and where conservation genetics and genomics seems to be going. I wanted to just start by saying start with the prelude here. Brad Schaefer, who used to be a faculty member here in evolution and ecology, is now down at UCLA. He directs the LaCrette Center for California Conservation. He does conservation genomics. He does workshops on this periodically. There's one early February in Sacramento. I don't know who the audience is for that, but anyway, he does these a couple times a year. Holly Ernest was doing more and more conservation in their applied ecology and genetics and genomics class. And well, she's gone now, but I believe the course still is on the books. So there's increasing level of towards genetics and genomics in terms of conservation in workshops and in courses. This figure down at the bottom is just sort of an indicator of publications that are, and the fraction of those studies that are using allozyme sort of markers, microsatellites, and other kinds of markers. And Jordan will talk about those. This is from Uberg's 2010 paper on thinking about the transition to conservation genomics from genetics. They'll talk a bit about at the end. So I'm going to start by thinking about how evolution is important in conservation, and there's a lot of different reasons, and I'm going to go through these all really pretty quickly. But humans are direct pressures. Humans act as drivers of undesirable evolutionary changes, and so we know this simply and specifically from the kinds of ways in which humans are drivers of changes that we find beneficial. So domestication of animals, and for example, is an obvious case. But we also know that whenever we put things into captivity, that we have a selective pressure that we maybe not intending. And so zoos and captive wildlife programs and botanic gardens all worry about selection to the captive environment, and that and loss of genetic material as a consequence of being in captivity. And so, for example, in zoos, as they move towards having more captive wildlife and participating in conservation, have breeding books and at least try and maximize the breeding around different zoos, hopefully maintaining more of that variation. We can also see examples of where they've tried to use zoo populations to actually create a change that you'd like. So, for example, Speke's gazelle was extinct in the wild, a very, very small captive population, and was experiencing inbreeding depression. And Fred Allendorf and others made the suggestion to drive inbreeding to try and call out some of these deleterious alleles. I believe that they felt that it worked at first, and then there was secondary skepticism about whether it actually worked or not. But people have used genetic principles in conservation of zoo populations for a long time. But fisheries management has an analog, that hatchery fish. We worry about hatchery fish and how they're responding to hatchery conditions in terms of selection. Aquaculture, same sorts of problems. But we can also think about this in the natural environment. So exploitation pressure causes selection. There's lots and lots of papers showing how fishing pressure in fished fish like cod or grayling or lobsters can change the minimum size to reproduction. And as a consequence, you're selecting for younger and younger reproduction, which reduces fitness because those animals are producing fewer eggs. And so it's not a favorable trait even for the fishery. This can also occur in hunting because of hunting pressure. And so Coltman et al. In 2003 showed undesirable evolutionary consequences of trophy hunting, smaller size, smaller antlers, and things like this. So human predation pressure causes variation in traits. And there's a paper in 2009 that human predators may outpace other predators as drivers of change. It's supposed to be change in prey. And so that's those are direct causes. But we also have indirect change. So human-driven environmental changes also drive evolutionary responses. So, for example, fragmentation creates risks in crossing inhospitable habitats such as roads, and there's then selection on organisms that cross roads better than others if there are behaviors that lead to more favorable or less favorable road crossing or in other inhospitable habitat crossings, just as wind turbines and other things are likely causing selection in birds now in terms of which ones are getting killed by those turbines and which ones aren't. But habitat loss also changes movement behaviors or selects for certain types of movement behaviors or certain types of propagule dispersal that can move from patch to patch. And similarly, that invasive species have been shown to drive evolution in native species. A great example of cane toads in Australia driving selection in their potential competitive species. And also then we have cases where invading species themselves are hybridizing with native species, the tui chubs in deserts of California and Nevada, the rainbow trout in the Sierras, California tiger salamanders are all local examples of these things, and there's been a pretty good literature on these. Scott Carroll has also looked at this with the soapberry bugs that have been introduced all over the world. And now on top of all this, we have climate change, and we worry about species and their capacity to change in response to climate change. In particular, we often worry that they're not able to change quickly enough. And so Richard Premack, and looking at phenological shifts in northeastern Florida, floras are looking at which species can and can't respond to the changing seasons, and we think of these as selective pressures. On the other hand, we might also worry that changing native species genetic composition in response to these anthropogenic environmental changes change these species away from our historical or natural in ways that we might not desire. So you can pause on a couple of these. I'm not going to go into too much detail on these. These are just two papers and a little bit of text on the hybridization event with the tiger salamanders and the tui chub, and then resulting in species or populations that we're not quite sure what to do with in terms of conservation. So anyway, there's this conservation double whammy that declining population sizes result in the loss of variability through random processes, these small population problems. But then changing environmental conditions can impose strong directional selection and environmental mismatches and drive further genetic losses. So we can either defend local adaptations and try and manage the change, or try and maximize genetic variation and to see that nature has something to work with. And here's kind of a manifestation of this of a sort. On the left of this graph, you'll see an old map. This is the California seed zones. And so foresters in California were instructed to replant forests with seeds from within the seed zone. And so you see California very finely partitioned up into these seed zones. You're not really supposed to move seeds outside that seed zone. Well, if you think that climate is changing, well then maybe these then become mismatched with their environment, and that's sort of hearkening to the historical. Starting in about 2005 and up through 2008, and this is from 2014, the chief forester of British Columbia has taken a completely different, has modified their approach. They had seed zones as well, and now here you see green, blue, and yellow, and these are the current ranges of the western larch and the potential future ranges with climate change. And the chief forester is now sanctioning planting species well outside their historical range, doing assisted migration in forestry, in effect, in order to maintain variability and maintain populations. And so this is an adaptation approach that they're taking, given the anthropogenic forcing of climate and the selection that may cause on species. Okay, so all of these problems with changing environmental conditions have greater potential to impact species with small population sizes, which means that it becomes then a big conservation problem. However, the thing I think that is most important to think about is that although it's a big conservation problem, it is one that often doesn't bubble to the surface, that I think it was Fred Allendorf a number of years ago said, well, look, when you're talking about conserving small populations, genetics may be important, but or that was Russell Handy who said this, but really the small population size, the allele effects, and the demographic stochasticity, these are going to outweigh the genetic problems 9 times out of 10. And for the most part, when we are talking about endangered species, people are having enough trouble trying to get anything done at the species level, much less worrying about the genetic composition of those species. And so we're not dealing with this too terribly much. Turning the attention now to thinking about genetic tools to describe variation, however, you can look at this problem from a different angle. So I think I mentioned the other day in class that Leanna Joseph and others, colleagues, did something very similar to what the Forest Service, the Fish and Wildlife Service has with respect to their priority system, although now they explicitly put in a species weighting. That's that W term here. So W is the species weighting, and they use taxonomic distinctiveness. The delta P is the benefit derived from reducing threat status. In other words, the magnitude of reducing the extinction risk. And C is the cost. And this then is a ranking for how you should deal with species. Well, so they use taxonomic distinctiveness. You could use phylogenetic distance. You could use lots of different things. And this really points us to the TUATARA and ENSATINA problem. And you could use lots of different examples, but I use like the TUATARA and ENSATINA because they're both, well, one's a lizard, one's a salamander. But there you see in the center diagram, the little green thing in the lower right, that's the TUATARA, and it has an inordinately long branch length when this group of lizards thrived and had their heyday more than 200 million years ago. Whereas on the lower left, you see that ENSATINA is this classic case of incipient speciation at all different stages. There's plenty of species or groups or lineages that are evolving faster than the ENSATINA, but it's just a nice one for comparison to the TUATARA. And so here are 2, 4, 6, 7 different species that are at some incipient stage of speciation, one from the other. This right-hand graph was left over from different slides. Sorry, it's basically saying if we're concerned about all levels of biodiversity, should we also be looking at hotspots for genetic diversity? And so, and so we can be thinking about what kind of attributes of the species that we're trying to target. Are we trying to target evolutionary potential or evolutionary distinctiveness? We can also be thinking about that at a landscape scale and thinking about where those places are. That's like the lower right graph up above on the haplotypes. This graph here is from a paper about Brazilian Atlantic forests, but thinking about phylogeographic history and the evolutionary potential of different places as a guide to think about where you want to do conservation. Okay, so I'm going to pause.